Welcome to Transhumanism Hub. What if aging isn't a ticking clock, but a set of biological instructions that may, under certain conditions, be rewritten? For centuries, we thought it was fate. Now, science is beginning to ask a very different question. In 2006, a scientist at Kyoto University named Shinya Yamanaka made a remarkable discovery. A living adult cell could be reset, not killed, not replaced, but driven back toward a state resembling that of an embryonic cell. His team exposed fully mature adult cells that had spent years performing specialized functions, to 4 transcription factors. These factors later became known as the Yamanaka factors. They were capable of dramatically resetting a cell's differentiated identity, allowing it to regain pluripotency and become what is known as an induced pluripotent stem cell, or iPSC, a cell capable of developing into many different tissue types. In 2012, the Nobel Committee awarded the Nobel Prize in Physiology or Medicine to Shinya Yamanaka. It took only 6 years from publication to Nobel recognition, making it one of the fastest awarded discoveries in modern biomedical science. But subsequent research began to shift the direction of the question. In 2020, a study from David Sinclair's laboratory at Harvard Medical School attracted widespread attention. Instead of fully resetting cells to a developmental 0 point, the researchers performed partial reprogramming. They reported restoring certain cellular functions and reversing some molecular markers associated with aging in eye tissue. In mouse models, animals that had previously lost visual function showed partial recovery of vision-related responses. At this very moment, every cell in your body carries a set of epigenetic information, a chemical archive that records the cell's state and history. Under the right conditions, some of the age-related changes contained within that archive may be partially reversible. Every cell in the human body contains 2 layers of information. The first is the DNA sequence itself, the genetic code. The second is the epigenome, chemical marks attached to DNA and its associated proteins that determine which genes are activated and which remain silent. As we age, these marks gradually drift. Genes that should be active become silent. Genes that should remain silent become active. Cells begin to execute their instructions less precisely than before. This is not exactly the same as DNA mutation. It is more like cells gradually losing the ability to read their genetic information correctly. These errors accumulate over decades and appear at different rates across different organs throughout the body. Current research suggests that Yamanaka factors can partially restore epigenetic information and reduce age-related epigenetic drift. The cells are not replaced, nor do they necessarily need to undergo extensive division. Instead, they are induced to recalibrate their gene expression programs. Cells do not know how old you are according to your passport or driver's license. They simply respond to the biological signals they receive, and under specific conditions, those signals can, at least in controlled laboratory settings, be partially rewritten. In Sinclair's experiments, researchers used only 3 of the 4 Yamanaka factors: OCT4, SOX2, and KLF4. They reported regeneration of optic nerve-related function and partial recovery of visual responses in mice suffering from simulated glaucoma and age-related degeneration. The cells within the optic nerve were not replaced, yet they showed signs of functional recovery. These findings provided some of the first evidence that age-related functional decline in mature neural tissue may not be entirely irreversible under experimental conditions. However, there is still a vast distance between the laboratory and the hospital. The challenge is not solely scientific, it is also regulatory. The U.S. Food and Drug Administration has traditionally evaluated therapies based on specific diseases rather than aging itself. Even today, aging is not officially recognized as an approved therapeutic indication. As a result, companies typically design clinical trials around conditions such as glaucoma, Alzheimer's disease, or cardiovascular disorders, rather than directly targeting aging reversal as a regulatory endpoint. This means that many reprogramming-related technologies remain largely confined to laboratories and preclinical development. At the same time, tools for measuring biological age have become increasingly sophisticated. Epigenetic age tests, such as the Horvath clock and Grimm age, have been able to estimate biological age from blood samples since 2013. A growing body of evidence suggests that the gap between biological age and chronological age is strongly associated with disease risk and mortality, although the strength of prediction varies across populations and study designs. In some studies, these measurements have even outperformed certain traditional clinical indicators in predicting long-term health outcomes. Several interventions, including regular exercise, caloric restriction, and high-quality sleep, have been linked to improvements in biological aging markers. However, these approaches generally slow aging rather than reverse it. Partial reprogramming aims for something more ambitious, restoring aspects of the cellular state itself. In 2026, a company called Life Biosciences advanced its candidate therapy, ER100, into a Phase 1 human clinical trial. ER100 utilizes the same OSK factors, OCT4, SOX2, and KLF4, while excluding c-MYC, which carries a higher risk of tumor formation. Researchers use engineered adeno-associated virus (AAV) vectors to deliver these genetic instructions into eye tissue, while doxycycline is used to control when the system is turned on and off. This approach does not alter the DNA sequence itself. Instead, it attempts to reprogram how cells interpret and utilize the genetic information they already possess. Before entering human trials, OSK had already demonstrated the potential to restore partial visual function and promote neural repair in mice and non-human primates. For this reason, the eye became the first organ selected to test the technology. For researchers, the most important goal of a Phase 1 trial is not to prove rejuvenation. It is to determine whether this form of cellular reprogramming can be implemented safely and controllably in the human body. Your biological age, therefore, is not a fixed number. It is a measurable state and potentially a modifiable one. Over the past 2 decades, scientists have demonstrated that epigenetic information within cells drifts over time, and that this drift may be reversible, at least to some extent. From Shinya Yamanaka's discovery that cells can be reprogrammed to experiments restoring tissue function through partial reprogramming in animals, and now to the first human clinical trials, the question has gradually shifted from whether this phenomenon exists to how it can be utilized safely and precisely. The eye will not be the final destination. It is simply the first organ chosen to validate the technology. If partial reprogramming ultimately proves both safe and effective in humans, future applications could expand to the nervous system, the heart, skeletal muscle, and many other tissues affected by aging. ER100 is only an early step along that path, but the deeper question it seeks to answer is whether humanity can learn to reset the cellular memory of age while preserving the identity of the individual. If you enjoyed this video, don't forget to like and subscribe. I'll keep bringing you more cutting-edge research in future episodes. See you next time!